Welcome everybody, and thank you very much for the kind introduction. As it was mentioned, I'm going to present some things about how we can employ cryo-electron microscopy and artificial intelligence to help visualize endogenous protein community members. But what are endogenous protein community members? First of all, I have to give you some background, and for this, I have to introduce our lab's methodology. In our lab, we study cell extracts because we believe that they bridge in situ structural biology with traditional structural methods, and we can employ with them all kinds of modern biochemical and structural techniques such as Cryo-EM, mass spectrometry, and of course a lot of computational modeling. Also, we have seen already that cell extracts, we expect them to retain principles of cellular organization. And after we have our cell extracts, we mostly focus in megadatton range protein complexes. Okay, so back in 2017, in a publication by my BI partners in the MBA, he had seen in his micrographs this type of peculiar organization of protein complexes. And they look very interesting because apart from proteins, we can see also microtubulins or RNAs that are helping stabilize these kind of interactions. And by acquiring some data sets back then, he could identify and characterize the fatty acid synthase at 4.7 angstroms as a member of these communities. In a previous work by our lab, we managed to, across cellular extract fractions, characterize two different complexes, but with mass spectrometry, but with negative stain EM and not cryo. And additionally, in previous works, we could gain deeper structural insights for a single complex at a time. So, with this work, we aim to go for more. How many complexes can we reconstruct and characterize from a single data set containing less than 3,000 parametric images? What is the maximum resolution we can reach for this? And can we achieve this de novo with minimum personal interference? To answer these questions, we went to our in-house glaciers, which has been working for us quite well, and we acquired the dataset, as I said, of less than 3,000 images, 2,100 to be exact. And in initial screening sets, before the major acquisition, we could again identify this type of community assemblies amongst proteins, with again some filamentous signatures over here, helping assemble all these together. After we see this, we once again go in our sample and biochemically characterize it. First of all, we fractionate our cell extract with centrifugal chromatography, and we focus on the fractions that are in the highest molecular weight, above 1 megadalton. Additionally, we use label-free mass spectrometry to characterize in-fraction content, and with in-house activity assays developed in our lab, we probe the cell extract fraction for activity for oxobutyrate hydrogenase complex and pyruvate hydrogenase complex. We also verify their presence with immunoblotting assays. The activity assays are quite important because they verify that in our fraction we still maintain an active state of our protein complexes, even after a single fractionation step. So, after a dataset, the analysis comes. And as I said before, we want to do this in as minimum perturbation from the user as possible. So we use block picking, and we have our initial set of particle picks, which we classified in 256 classes, and immediately we could see 4 signatures popping up in the class averages at the highest resolution. We could see this cubic signature, this circular polyhedral signature with this diffuse density ring on the periphery. We could see this dihedral signature, and also we could see this completely asymmetric signature. So we grouped similar signatures together, and then we proceed for the next parts of the image analysis. What is important to note here is that, for example, even though the classes look quite high resolution, for signature 1, for example, we only had 1,800 particles to work with. From the dataset that contained in the beginning picking steps, more than 250,000 particles. So, we take our particles, we do ab initio reconstructions, and now we can recapitulate these peculiar shapes in 3D. And once again, we see for signature 1 this cubic shape, for signature 2 this circular ecospherical shape, dihedral for signature 3, and for signature 4 completely symmetric. And now we have the tough job to identify what the signatures are. And the thing is, as we're using a cell extract, we're not always sure what exactly is in there. And this is why we do mass spectrometry to identify the infraction context. And then we have another step that we can combine with to lower down our possible targets. In this case, we use the online database called Omokage, which is based on shape similarity searches, and you can blast your maps against the complete EMDB and PDB and gain a correlation coefficient score based on similarity of the target hits. And after doing this for all signatures, for signature 1, we get back 2 possible hits, the branching ketoacid dehydrogenase complex and the exoglutarate dehydrogenase complex. And these are also in the mass spec, so we have further verification. For signature 2, it was completely unambiguously characterized as the A2 core of the pyruvate dehydrogenase complex. Fungal fatty acid synthase was identified for signature 3, and for signature 4, the top 10 ranking hits were quite heterogeneous. And in order to tackle this, we 2D projected our own reconstruction and the hits returned by the database, and we compared them in 2D. And because of heterogeneity, it was very simple and easy to identify that our signature 4 belongs to the 360S ribosomal subunit of Hetomium thermophilium. After we have identified our particles, we can proceed with high-resolution reconstructions, and we could see in all cases some high-resolution features. In the case of the fungal fatty acid synthase, we could see chemical bundles. Again, we could, for oxoglutarate dehydrogenase complex, for branching ketoacid dehydrogenase complex, we could recapitulate this diametric interface on the periphery. For pyruvate dehydrogenase complex, the resolution was high enough and we could see side chain densities and beta strand separation. And finally, for the 360S, we could see the chemical bits of the RNA components. These are the quality metrics for all 4 reconstructions. You can see that the resolution distribution is quite decent for all 4, especially high resolution for signature 3 for the pyruvate dehydrogenase complex. And we could also identify the high flexibility features, as is in the case of fatty acid synthase, these peripheral subunits, and for the 360S ribosomal subunit, the flexible RNA components on the periphery. After we have our reconstructions, we can go and further investigate to acquire high-resolution insights, structural insights. And in the case of the 360S, we could fit various already pre-published pre-60 RNA, pre-60 ribosomal subunit components and identify 23 ribosomal subunits that are taking part in ribosomal biogenesis. And we could also see that one of the higher-order structural features that are formed during the ribosomal biogenesis is the protein exit tunnel. So, as I said earlier, in the case of signature 1, we had this issue. We could not identify whether it belonged unambiguously to either branching ketoacid dehydrogenase or pyruvate or exoglutarate dehydrogenase complexes. And for this, to solve this problem, we collaborated with Grzegorz Szanowski of EMBL Hamburg, who has developed various computational tools. He was one of the developers of ArcWarp, for example. And he recently published a very interesting tool called Find My Sequence. And we employed this in this pipeline for unambiguous identification. And how this starts is we take a native cell extract map, we predict all possible targets with AlphaFold, and then we define these targets in our original map, experimentally determined. We extract the backbone, and then Find My Sequence finds the correlation between the extracted backbone and our experimentally determined map. And after using this, we could unambiguously identify that indeed our map belongs not to the branching ketoacid dehydrogenase, but to the exoglutarate dehydrogenase complex core. So we're done with identification, and now we can also gain some very interesting insights on how AlphaFold predicted models can differentiate from models that have been refined in our maps. The insights concern the OGDHC, autoxylated hydrogenase complex, the fatty acid synthase, and the pyruvated hydrogenase complex, P2 course. And we'll start with the pyruvated hydrogenase complex. From the start, even from the map itself, we could see some additional densities on the inside of the core that we could not previously recapitulate in a previous publication. And these are interesting because they probably belong to another protein that's in the core, for which we have already have another publication where we identify these inside densities. But what is more interesting is that the alpha predicted model has this primary interface of alpha helices at the end-terminus, predicted to be ordered and backfolded into the trimer. And the end-terminus of each of the subunits of the trimer is very flexible and has lipoid domains and peripheral subunit binding domains that control the availability of the peripheral subunits that are taking part in the pyruvate reaction. But the thing is that in our experimental data, these helices are missing. And we see that these helices are blocking the accessibility to the CoA binding domain. Hence, we can see that alpha predicts an inactive state of the complex, whereas we have verified with our activities that in our fraction, our complex is active. In the case of fatty acid synthase, we could compare directly our map with the previously determined map, and we could see that we have now an external subunit in the middle over here that was not previously characterized, and we have found it to belong to the PPT CoA binding domain of fatty acid synthase. And what is also interesting to see here is that in comparison to the yeast published data, our information of this subunit is more open and varies by 25 degrees compared to the yeast model. And this is partly attributed to the more flexibility that is at the connector of this domain, and we believe that it helps maintain higher accessibility for the CoA to bind there and continue the reaction. Additionally, for the CoA, when we see the alpha versus the experimentally defined model, what was very, very interesting was that alpha recapitulated de novo this hexamerization interface at the center of the complex. These are two very extended helices that are connected by a loop and have this gamma conformation, inverse gamma language, and they help assemble the whole complex. And alpha, without any kind of previous knowledge, could recapitulate this kind of forming. Of course, it looks much healthier when we refined our map, but still it was quite a surprising result to see. And additionally, alpha for the PPT CoA binding domain predicted the same type of orientation as in the yeast and not as in our experimental observations. Finally, for the autoxylated hydrogenase complex, here you can see how we could not differentiate initially between OHDHC or BCKDHC. And because the overall shape is quite similar, and by fitting the densities to each other multiple times, the distribution difference was not significant. And this was what had the need for this development of the certification pipeline. And as for the alpha versus the refined model, once again, alpha predicts these lipoid domains that are transferring the reaction intermediates from one subunit to the next as permanently bound to the trimer of the core. And once again, we see these missing from our experimentally defined map. And if they were permanently docked there, this once again would annotate an inactive state. And we have verified, first of all, that with activity assays that our autoxylated hydrogenase reaction is happening, so our complex is active. And we could also see here the 2D class averages. Of course, the central density is very robust, very stable, but also we could see a cloud of very diffuse density on the periphery, also giving more value to the flexibility and high movement of this lipoilar on the peripheral protein subunits. So, to conclude, we employed cell extracts to investigate protein community members, and we could reconstruct and identify 4 different signatures that belong to members of the communities. We used as unobtrusive sample preparation as we possibly could, and Cryo-EM data collection modeling schemes. We complemented biochemical assays, mass spec, statistical analysis, and other bioinformatic methods to validate our results. And we can see that the newest advances in AI and Cryo-EM image analysis and de novo model prediction can further this type of structural knowledge. Another interesting take-home message is that AlphaFold-generated models are quite robust, they're good, but they should not be taken for granted. It's good to have a starting point, but ideally you should refine them or at least gain some more insights concerning for them from your experimental data. Okay, so this work comes to complete the picture that our lab wants to present. In a semi-reports paper in 2021, we established our lab's workflow to work with cell extracts and investigate proteins in the native state. And we validated this with biochemistry, and we got to propose a model for the organization and activity of the pyruvate hydrogenase complex, the pyruvate hydrogenase factory. Now, this structure paper comes to as the second part that gives us more insights on other complexes that are in there and what we can do concerning cell extracts, how we can analyze them. And finally, we also have a publication in Nature Communications last year that delves deeper and gains higher resolution insights into the pyruvate hydrogenase core itself and completely describes the acetylase reaction. And I was also very happy to polish my graphical artist skills. You know that as PhD students, we have to do this. I think it's a trick to, if we are jobless afterwards, we go into graphical artist businesses. Yes. And also the Ioannis group in Münster wrote a very nice review for our work, and we thank them a lot for this. And at this point, I would also like to thank you for your attention and the organizers for giving me this amazing opportunity to present my work to all of you. Thank you.